Good morning. My name is James Polli. I'm a faculty member at the University of Maryland, and welcome to the FDA CRCG workshop on formulation characterization and cutaneous pharmacokinetics to facilitate generic topical product development. So the purpose of today's workshop on formulation characterization and cutaneous pharmacokinetics is to discuss efficient science-based BE approaches in development that may be useful for certain prospective topical generic products that are compositionally different compared to the reference standard. Slides and recordings of today's workshop will be available in about 3 weeks, and all of you, all the registrants, will be emailed when they're available. So be lookout for an email in about 3 weeks' time. Agenda and speaker biographies are also available at our website, complexgenerics.org. And let me just show you what they look like. Now is probably a good time to actually take a look at those documents. There's 2 of them. Here is the agenda for today. It's a 4-page document. And this morning we have some introductory comments from Dr. Luke and Dr. Rainey, followed by a brief coffee break. And then we have 3 sessions, one in the morning regarding understanding the influence of formulation differences on product performance. And then after lunch break, session 2 is on development of cutaneous PK-based approaches, followed by the 3rd session on method development and validation of cutaneous PK approaches. Okay, so I encourage you to, and if you can kindly look at the biographies, we'll be doing brief introductions through the course of the day. Just a little bit about the Center for Research on Complex Generics. CRCG, the mission is to increase access to safe and effective generic products through enhanced infrastructure and communication, education, and research collaborations across academia, industry, and the FDA. I would encourage you to learn more about the center by going to the website, complexgenerics.org. And you can also sign up to receive an email via our listserv. We send about 1 email a month. We try not to send too many out. But by signing up, you'll learn about things like, for example, currently we have a survey on best practices and standards in nanotechnology. Underpinning this survey is the desire to facilitate the development of new and generic complex products, including nanotechnology-based medications. And that survey is open through a week from now. We do have 1 additional workshop, an online workshop later this year in December, about a month from now. It's more of a training, and it concerns excipients and formulation assessments of complex generic products, best practices, and lessons learned. So recently we announced a lineup of 6 in-person and online workshops, so hybrid, so to speak. These 6 workshops will be held at the University of Shady Grove in Montgomery County, Maryland, just north of Washington, D.C., in Rockville, Maryland. The topics for these 6 workshops are on drug-device combination products, sort of a tutorial training. That's in March, so that's our first in-person workshop. In April and June, we have workshops on orally inhaled drug products, as well as nitrosamine drug substance-related impurities. And then next fall, we have a workshop on physiologic-based pharmacokinetic modeling for all drug products, one on drug-device combination products, a more specific topic regarding comparative user interface assessment, and then finally, to round out 2023, complex excipients and formulations. So thank you very much for your participation. Please ask questions via the chat. I know the moderators will be working hard to try to answer all questions today. And again, just a final note, slides and recordings of this workshop will be available in about 3 weeks, and you'll be emailed with regard to when they become available. Okay. And it is my pleasure to introduce Dr. Markham Luke, who will be providing introductory comments about clinical relevance. Dr. Luke serves as the FDA supervisory physician and director of the Division of Therapeutic Performance 1 in the Office of Research and Standards in OGD at FDA. The Division of Therapeutic Performance 1 is responsible for facilitating pre-application development of generic drugs by conducting and promoting regulatory science research to establish standards to ensure therapeutic equivalence of new generics. Markham has research interests in dermatopharmacology, clinical study design, and endpoints assessments for medical, surgical, and aesthetic products, and it serves as a consultant dermatologist to various parts of the FDA. Markham has been at FDA since 1998. Thank you for your introductory comments, Dr. Luke.
Thank you, Jim. Thank you for your kind introduction. And it's a pleasure and honor to participate and speak at this Center for Research in Complex Generics workshop. And I just point out this is a workshop, and so we welcome comments in the chat room and throughout this session. And also I want to put in a plug before I start my talk for what a great place FDA is to work. And we are looking for folks who have an interest in dermatopharmacology or pharmacology in general. Please send your CVs. If you're a PhD candidate approaching your graduation year, we have fellowship opportunities, and there are job opportunities at FDA. So I wanted to go into this talk. My talk is about understanding the clinical relevance of formulation for topical products applied to skin. Human skin, as most of you know, is a complex entity. It has many functions in addition to its barrier function, including temperature regulation, providing tactile temperature, vibration sensation, moisture preservation, protection from solar radiation, and providing protection from microorganisms. Also of interest to us is that the skin is an outward projection of who we are. It is one of the human organs whose appearance has overt cosmetic, cultural, and societal implications. Skin has multiple structures relevant to its many functions in addition to the layers of cells and scaffolding and stratum corneum. These include follicular units, which include hair, root sheath, associated glands, nerves, muscles, and stem cells. It also includes underlying blood vessels, smooth muscle, underlying fat, structures, and skin appearance may vary depending on where on the human body we're looking at. As a licensed to practice dermatologist, I can appreciate the relevant role these structures or damage to these structures may play in disease and normal skin health for my patients. I can also appreciate how these structures impact the delivery of drug to the putative sites of action for drugs we use to treat various skin diseases or conditions. So I wanted to just go over some of these. What are some of these diseases? Acne, psoriasis, eczema or atopic dermatitis, cold sores, alopecia, rosacea, skin cancer. These are just some of the examples. Some of these disorders have effective systemic drug and biological therapies developed recently. However, topical drugs continue to play an important role in the integrated holistic management of these diseases. And hence our interest at FDA in developing good topical drugs, both new topical drugs and generic topical drugs. What aspects of product formulation impact drug delivery and in turn impacted by skin structure? The active pharmaceutical ingredients, API, key issues are dissolution and the size of the API particles. The particles themselves participate in drug release functionality for the product, but also allow for API in its undissolved state to penetrate and accumulate, say around hair follicles and other dermal substructures when relevant. This is especially relevant, for example, in acne or to treat perifollicular inflammation. And this is something to keep in mind about site of action for products. The excipients go hand in hand with each type of API. For example, hydrophobicity, and the target indication. The clinical mantra of if it is wet, dry it, if it's dry, wet it, is often addressed within the formulation of drugs used to treat the target disease with appropriate excipients. These excipients play an important role in how the product functions. It should be noted that for topical products, excipients are often misidentified as inactive ingredients when they play a crucial role in the treatment of disease, including substituting as a barrier for broken skin, reducing water loss, providing a reservoir environment for the API, performing penetration functions for both drug and co-solvents, providing relief to irritation and itch, and improving the spreadability or look and feel of a product applied to the skin. Over the centuries, substitution of excipients that perform certain functions has occurred with benefit to users. For example, substituting certain fatty acid ester mixes like crotamol or PCL liquid for the natural preen oil, sparing source animals and making certain topical products friendly to consumers with certain beliefs, for example, or substituting one thickening agent or emulsifier for another. In the context of generic drugs, this substitution should be done keeping Q3 parameters and drug availability intact. And we're gonna have a little bit of discussion during this workshop about Q3 and the importance. And I also wanna take this opportunity to highlight the recent release of our Q3 guidance. It's a draft general guidance on physical, structural, physical, chemical properties for topical products. This is long in the coming. It's been about a few years that we've been trying to work on getting this out in form for public consumption. So please do, folks who are participating in this workshop, please do read through that, look to see if you have any questions, ideas, comments, send them to us to the docket, which is open in conjunction with the Q3 guidance. We welcome those and look forward to hearing from you regarding our Q3 guidance. In the context of generic drugs and comparing a test product to the reference product, the topical drug field has evolved over the course of the last 3 decades, from initially having in vitro comparison of Q1 or qualitative comparison, that is the same ingredients considering grade and morphology of the ingredients being put into the formulation, Q2 or quantitative comparison, for that is the same relative amount of each ingredient, which might have some variation due to measurement capabilities or variation in batches, and adding in Q3 or physical, chemical, and structural differences evaluations in the early 2000s. For example, rheology, API characteristics in a manufactured product is applied to skin, pH, spreadability, etc. During the course of this workshop, you'll hear more about some of these Q3 parameters, which have become important in the measurement of in vitro characterization. The keys to matching Q3 are considerations for variations in Q1, possible slight but allowable differences in Q2, and most importantly, manufacturing and packaging processes that lead to the final marketed formulation. Differences in manufacturing can include intensity of mixing, temperature at which mixing is done, the order of addition of the excipients and API, the introduction of unique aspects that could impact final physical chemistry and structure, API bioavailability, and the look and feel or sensorial attributes of the final product. These are some important considerations for developers who want to develop topical generics, and it's something for FDA regulators to keep an eye on as applications come in. For indications, so I'm going to go into a little bit about some of the disease indications, how the Q1, Q2, Q3 differences might impact some of the products. For indications like acne vulgaris, the site of action may be at the root of the hair follicle, that is providing antimicrobial, anti-inflammatory, and anti-keratinization, thus requiring API access to the site of action. In general, matching Q1, Q2, and Q3 will help us arrive at a bioequivalent test product. As we develop the science of understanding the relevance of certain Q3 attributes, this may evolve. For example, particle size may not need to be entirely matched, but could possibly be related to having a proportion at a certain size cutoff. in a certain localized concentration to allow the drug product to work effectively. Currently, we don't know what that size cutoff is. There may be further complicated by the need to reduce adverse events such as irritation, which could have an adverse impact on the frequency of application and patient dosing. For these reasons, at this time, an adverse impact on frequency of application and patient dosing. So for these reasons, at this time, if there are variations in Q3, clinical endpoint studies may still be needed. And so it's important to keep that in mind. And clinical endpoint studies continue to remain an option. In our product-specific guidances, we highlight, yes, we think sometimes that the in vitro approach is the way to go, but we continue to want to hear from you if you're developing a clinical endpoint study and you think that you'll have greater success from that. We're interested to work with you on getting those studies underway. For indications like cold sores, it may be the film forming ability of the formulation that allows for a therapeutic protective layer over an open sore. For diseases where chronic response to inflammation might be stratum corneum thickening, it may be important to have sufficient skin penetrating solvent to carry dissolved API. A Q3 approach, together with dissolution, IVRT, and skin penetration assays like IVPT, may be sufficient as proof of bioequivalence or BE. So these are some important considerations to keep in mind from a disease perspective. For topical dermatologic products, it's not always necessary legally, regulatorily, or practically for products to be Q1 and Q2 to have the same bioavailability or same performance as has been shown in the past with approved ANDAs, some of which have utilized a comparative clinical endpoint approach to BE. So we've approved ANDAs that have formulations that are not Q1, Q2, and that's okay. Q3 sameness may at this time in our generic drug regulatory evolution be a stand-in for comparable performance. But as we learn more about what Q3 aspects are necessary for each target indication, the relevant sets of data will be tweaked. So I did want to make sure that if folks have a bit of time for questions, I'm happy to take those. But in summary, for this talk, it's important to understand the disease and skin location to which the product is applied and the site of action of the drug under consideration. With appropriate application of analytical and measurement science, we can get to yes for BE in more ways that are valid, appropriate, and clinically responsible. And with that, that ends my fireside chat with you all. And look forward to hearing any comments or questions. Thank you.
Okay, thank you very much for those introductory comments, Dr. Luke, about clinical relevance. And I should have added, those of you who have long practice topical product development probably know Dr. Luke and maybe recognize him from professional meetings where he's often seen with a camera. If you've practiced in topical product development for a while, you also probably know also Dr. Sam Rainey, who will be providing introductory comments about GDUFA research efforts. Dr. Rainey is a thought leader in topical and transdermal product drug products with over 30 years of experience in skin research, producing numerous research manuscripts. He's the Associate Director for Science at the FDA's Office of Research and Standards and serves as a Chief Scientific Advisor for topical product bioequivalence issues in OGD. Thanks for your introductory comments about GDUFA research efforts, Dr. Rainey.
Thanks so much, Dr. Polli. And Dr. Luke, that was a really nice way to start our workshop today. So thank you so much. I would like to talk a little bit about the strategic research initiatives that we've been engaged in for the better part of the last decade, focused on developing efficient bioequivalence approaches for complex generic topical products. And I'll offer the standard disclaimer here, but begin with our Code of Federal Regulations. These are really what guide the way that we regulate topical products. So just some basics to begin with, that a generic drug product must demonstrate that it's bioequivalent to the reference listed drug product, and that's typically done by demonstrating bioequivalence to the reference standard, the designated reference standard drug product. That's the product that's available on the market and designated as the reference standard. Sometimes it is the original reference listed drug product. And bioequivalence is defined in our CFR as the absence of a significant difference in the rate and extent to which the active ingredient becomes available at the site of action. Now, the type of evidence that you can use to demonstrate this is also described in our CFR. This is 21 CFR 32024. And the evidence that's considered to be the most accurate, sensitive, and reproducible for determining bioavailability or bioequivalence is an in vivo test in humans in which the concentration of the active ingredient in whole blood, plasma, serum, conventionally these are plasma PK studies for solid oral dosage forms, or other appropriate biological fluid is measured as a function of time. So these are pharmacokinetic studies. Now, if we look at the same CFR and we kind of remove some of the interceding language here in terms of how does this apply to topical products applied on the skin, what this basically indicates is that an in vivo test in humans in which the concentration of the active ingredient in an appropriate biological fluid, let's say dermal interstitial fluid, is measured as a function of time, that would provide the most accurate, sensitive, and reproducible evidence for bioequivalence. In other words, a cutaneous pharmacokinetics or cutaneous PK-based bioequivalence approach is really considered to be the ideal according to our regulations. And several such approaches have been conceived over the years, including tape stripping approaches or dermal microdialysis and dermal open flow microperfusion, as well as non-invasive spectroscopic methods that are sort of like a medical tricorder. They can non-invasively detect the concentration of drug in the skin over time and see how that's changing and compare that for products. Now, there have been some fundamental scientific and practical challenges with developing these approaches, including challenges relating to how you get to the same sampling depth, or the frequency of sampling, the duration of sampling, a lot of issues with reproducibility and variability, as well as the fact that the fundamentals, the scientific fundamentals of pharmacokinetics for cutaneous pharmacokinetics has some nuanced differences compared to systemic pharmacokinetics. But for decades now, FDA has supported research to develop these types of approaches. So the focus is really on developing efficient approaches for topical bioequivalence. And so in parallel, FDA also supported research to develop in vitro bioequivalence methods. And in fact, that same CFR that I referred to before, 21 CFR 32024, also lists in vitro evidence that can demonstrate bioequivalence. It talks about how an in vitro test that has been correlated with and predictive of human in vivo bioavailability data, or a currently available in vitro test that's acceptable to the FDA. Now, for solid oral dosage forms, for example, that would typically be a dissolution test that ensures human in vivo bioavailability is also an appropriate type of evidence. Now, for topical products, these in vitro BE approaches may include an in vitro release test, which is sort of the corollary to the dissolution test for solid oral dosage form, as well as it may include an in vitro permeation test. So these IVRT and IVPT tests have been very valuable. And notably, these IVPT studies are also cutaneous pharmacokinetics-based BE approaches. It's just that they're in vitro. Now, why is this relevant to today's workshop? Because numerous of the scientific advances that supported the regulatory utility of IVPT studies also laid the foundation to make in vivo cutaneous PK approaches feasible and more efficient. This includes study design considerations, a lot of the approaches to validation, as well as data and statistical analysis methods. And we've really been focused on modernizing topical bioequivalence. There have been two strategies that we've utilized. One strategy facilitated the development of topical generics that were precisely matched to the reference standard, within batch-to-batch variation of the reference standard product. Now, how could you figure that out? How could you tell that it was precisely matched? Well, we had to develop some tools. And to assess this, we supported research that developed and optimized some in vitro tests to compare the physical, chemical, and structural, the Q3 attributes, the product quality attributes, as well as IVRT bioequivalence studies to compare the rate of drug release from the dosage form, and IVPT-B studies to compare the rate and extent of drug permeation into skin. So this strategy was really focused on modernizing topical bioequivalence by developing an efficient approach where you could demonstrate that the generic product was identical to the reference product, as identical as batch to batch of the reference product was to itself. And therefore, for the same reasons that you wouldn't re-demonstrate bioequivalence in some clinical study or therapeutic effectiveness in some clinical study every time you made a new batch of the reference product, likewise, because the generic product was so precisely matched, it was implicitly bioequivalent as well, because it had systematically eliminated any kind of mitigated the risk of failure modes for bioequivalence. So this was one strategy. Now, another strategy that facilitated the development of in vivo cutaneous PK-based BE approaches. This is precisely aligned with the CFR for the types of evidence that's ideal here. And this included in vivo dermal open flow microfusion or DOFM studies and dermal microdialysis or DMD methods, which are currently in an advanced state of development and is now one of the tools that we're developing in the toolkit of methodologies that have been supporting these two major strategies. So currently our topical bioequivalence toolkit includes multiple complementary approaches to address failure modes for bioequivalence. Failure modes being things that a difference in a generic product might cause to make it non-bioequivalent. In other words, we want to be able to understand the complexity of each topical reference product, match that to, or map that to potential failure modes for BE that can arise from differences in the product, and then systematically mitigate the risk of those failure modes for BE. So the toolkit included tools to establish when there's no significant difference in Q1 or Q2 attributes about the ingredients, the qualitative or quantitative composition, tools to compare the Q3 attributes between two topical products, and to perform IVRT studies, including tools for method validation, BE study conduct, and data analysis, similarly for IVPT, as well as tools to perform in silico modeling and simulation that could support a demonstration of bioequivalence. And of course, we had well-established tools for in vivo systemic, this is plasma PKB studies, which also sometimes can be used to support a demonstration of BE, as well as, in addition to the systemic or plasma PK studies, the in vivo local or cutaneous PKB study conduct and data analysis that's also part of the toolkit that we're developing. Now, if you're a product developer, one of the ways to think about how you can implement efficient topical BE standards, there's two major pathways that you can think about. One is that for product formulations that have no significant difference, this is the standard for Q1 and Q2 matching, no significance different than Q1, Q2, or Q3 attributes compared to the reference standard. These products are typically eligible for in vitro-based BE approaches. Whereas product formulations that have a difference in Q1, Q2, and Q3 attributes are typically eligible for in vivo-based BE approaches. But let's unpack that a little bit. So we've recently released the Q3 guidance for industry. It's a draft guidance. And one of the ways that it encourages you to think about pathways is to ask the question first, is the arrangement of matter the same between the test and the reference product? This refers to the Q3 attributes. If no, then it's a Q3 different product. You have a Q3 difference. If the arrangement of matter is the same, all the Q3 attributes are really well matched, then yes. Then the next question is the underlying matter, the Q1 and Q2 attributes, are they also really well matched? In which case you've got the same arrangement of the same underlying matter, and there you have Q3 sameness. But there is another situation where you can end up having the same arrangement of matter. So all the Q3 attributes match, but there is some difference, potentially significant difference in Q1 or Q2 attributes. So you have the same arrangement of slightly different underlying matter. And this is Q3 similarity. Now, this framework for thinking about things affords pathways for approval of products that we're going to explore during today's workshop, particularly during session 1. Another way to think about this is that we have product-specific guidances. And these product-specific guidances will typically guide that if you have a Q3 different product, then those products are generally eligible for traditional in vivo bioequivalence approaches that are currently recommended in PSGs. There's a comparative clinical endpoint bioequivalence studies, as well as perhaps vasoconstrictor studies in certain cases for the corticosteroid products. If, on the other hand, you have a Q3 product, product that's Q3 the same, these are generally eligible for the characterization-based in vitro BE approaches that are currently recommended in PSGs. But we don't currently have any PSGs that recommend what to do in the case of a Q3 similar product. And this is partly because there are so many different ways that you could end up creating these differences, and those don't have the same effect. Some changes will have a very modest effect. Some changes may have significant, a significant and very different failure mode for bioequivalence. And so the approach here is to understand what that difference is and what's the failure mode for bioequivalence that's implicated, and then to think about what's the type of evidence that would say, well, in this case, we are mitigating the risk of that potential failure mode for BE by providing that type of evidence. And maybe that evidence will be, it's a product that the PSG, if it was Q3 same, would say you do Q1, Q2, Q3 IVRT, and now you add on an IVPT study. Or maybe you would use modeling and simulation to demonstrate that there's no real risk for that potential failure mode for BE. But in many situations where these types of in vitro or in silico approaches may not be sufficient, it may be very useful to utilize in vivo cutaneous PK like the DOFM and DMD-B studies. And that's a big part of what we're going to talk about today. So where are we at with these studies? Well, the current state in terms of their benefits and the challenges and the opportunities are that, as we talked about from the CFR, this is an ideal type of evidence to demonstrate BE for topical products. We've had multiple drugs and multiple dosage forms studied by multiple researchers. So this is now, very clear. This is a very good generalizable approach for many topical products. Key elements of protocols and operating procedures have been developed, and a lot of the considerations for the study designs, the controls, and the validation are well defined. So the scientific principles for cutaneous PKB studies are in an advanced state. However, study design elements and analytical methods will need some optimization, and data and statistical analysis procedures may also need some optimization. We continue to have research over the next couple of years looking at these. That's why we wanted to engage all of you in today's discussion. And the goal of today's workshop in these sessions is that each of these sessions is intended to help us discuss, one, the influence of formulation differences on failure modes for BE in vivo, and potential contexts of regulatory use for in vivo cutaneous PKBE studies. We'll also look at the scientific and practical challenges with conducting and assessing these in vivo cutaneous PKBE studies, and the considerations impacting their efficiency in particular, as well as the specific procedural and technical methods that we can optimize to ensure that these in vivo DOFM and DMD studies efficiently mitigate those failure modes for BE that can arise with Q3 similar products. So the goal of today's workshop is to identify scientifically appropriate, practical, and efficient approaches for conducting and assessing these in vivo DOFM and DMD-B studies that generic product developers hopefully would likely consider to be feasible to utilize for the development of topical generic products that can enhance patient access to these really important medicines. So with that, I'll say thank you so much. There are so many people who have contributed to this work over the years, and a few of them who are mentioned here have specifically contributed to this presentation. With that, I'll say thank you so much and look forward to connecting with you during the sessions of our workshop today.